We are now going to talk about what is perhaps the most important macromolecule in life, and that is known as nucleic acid. Now, first of all, where does that name come from? Well, scientists first observed this in the nucleus of cells, and so that's where you get the nucleic part, and it has some acidic properties, and so that's where you get the acid part. And perhaps the most famous of the nucleic acids is deoxyribonucleic acid, or DNA for short. And we'll go into some depth in this as we go through our journey in biology, but you might already know that this is the molecule that stores our hereditary information. This DNA, to a large degree, makes you you. And it's known as a macromolecule, and we've talked about macromolecules in other videos. We've talked about carbohydrates, and we have talked about proteins. And DNA is a macromolecule because it can be made of many millions of atoms. Just to get a sense of it, you can see right over here the double helix of DNA, where you have one side of your helix right over there, and then another one right over here, and then you kind of have these rungs of this twisted ladder. A DNA molecule, let's say in the human genome, a chromosome, for example, is primarily a really long DNA molecule. And it can have on the order of 100 million rungs to this ladder. Now, another thing to appreciate, like many other macromolecules, DNA, or nucleic acids in general, they are polymers, in that they are made up of building block molecules. And those building blocks for nucleic acids, and DNA is the most famous nucleic acid, and RNA, ribonucleic acid, would be a close second, but the building blocks of them are known as nucleotides. Nucleotides. And we see some examples of nucleotides right over here. This is deoxyadenosine monophosphate, which would be a nucleotide found in DNA. You can see the various parts of it. You have a phosphate group right over here. You have a 5-carbon sugar, which in this case is ribose. And then you have what is known as a nitrogenous base. And why is it called nitrogenous? Well, those blue circles represent nitrogen. And we've seen this before. The grays are carbons, and the reds are oxygens, and the whites are hydrogens. And so this part of the molecule has some basic characteristics, while this phosphate group at the end, this has some acidic characteristics. And what happens is they get stacked onto each other, where the ribose phosphates alternate to form the backbone of this DNA molecule. You can see it right over here, where you have a phosphate and a ribose and a phosphate and a ribose. And then you have the nitrogenous base forming part of the rung of the ladder. And the way that DNA stores information is every one of these nitrogenous bases right over here, this is adenine, it has a complementary nitrogenous base on the other to complete that rung of the ladder. So adenine matches with thymine in DNA, and we'll see in future videos in RNA, it's a nitrogenous base known as uracil, and guanine matches with cytosine. Just appreciate that the monomer for a nucleic acid like DNA is a nucleotide, so monomer. And to be very clear, this would not be the only monomer. The analogous nucleotide in RNA, which stands for ribonucleic acid, would be adenosine monophosphate right over here. You can see the difference between the two, that we have an oxygen right over here and we don't have an oxygen right over here. That's why this is called deoxy, and that's why it's deoxyribonucleic acid. You're missing one of those oxygens on your 5-carbon sugar. But adenine, as I mentioned, is not the only nitrogenous base. You could have a nucleotide where the nitrogenous base is thymine. And so once again, this looks very similar, but notice what is going on over here. You could have a nucleotide that looks like this. Once again, you have your 5-carbon sugar here, you have your phosphate group, but the nitrogenous base here keeps on changing. And it's the order of these different nucleotides that actually encodes the information in DNA.
Nucleic acids are basically biopolymers which are made up of monomeric units known as nucleotides. We have two important functions for nucleic acids, that is, they hold our genetic information and they perform a variety of other functions that we will discuss later. We have two important types of nucleic acids in our body, the DNA and the RNA. Let's first try to understand the definition of nucleic acids. So nucleic acids are biopolymers. Bio means that they are synthesized inside biological systems and they are not synthetically synthesized. They are polymers, which means they are made up of repeated units called as monomers, which in this case are nucleotides. Now take this example. We have many different types of biopolymers in our body. One of them are the proteins. And most of you know that the proteins are made up of repeated units called as amino acids. Similarly, nucleic acids are also important biopolymers in our body, which are made up of repeated units which are known as nucleotides. So the next important thing is to understand the basic structure of a nucleotide, which will help us to understand the structure of nucleic acids in detail. Now this is a simple diagram explaining the structure of a nucleotide. As you can see, the single nucleotide is made up of three important groups: the phosphate, the sugar, and the nitrogenous base. And these groups are attached to one another by bonds, which we'll discuss later. To understand the complete structure of a nucleotide, let's try to understand the structure of these groups one by one. So the first is the sugar. The sugar present inside the nucleic acids are 5 carbon sugars. Now most of you know the 6 carbon sugar present inside the body, and most important of them is the glucose. Now in chemical nomenclature, all the 6 carbon sugars are called as hexoses. The word hex meaning 6. There are different types of hexoses in our body other than glucose, like the mannose and galactose. Similarly, 5 carbon sugars are called pentoses, the word pent meaning 5. There are different types of pentoses in our body, but the two important types of pentoses that are present inside the nucleic acids are the ribose and the deoxyribose. Now this is a simplified structure of the sugar present inside the nucleotide. Now if we compare this structure with the chemical structure of the ribose and deoxyribose on the right, you can see the structure clearly. We have carbon atoms on each of these corners. Note that the last carbon atom is located outside the ring. We have an oxygen atom at the center topmost position. You can also see in this chemical structure that the carbon atoms are named from 1 to 5 in a clockwise direction. So the carbon atom next right to the oxygen is called C1, so C2, C3, C4 and C5. C5 is located outside the ring. You can also see in this chemical structure we have different type of functional groups attached to these carbon atoms, like the hydroxyl group and the hydrogen atom. The most important of these functional groups, which determine whether the sugar is ribose or deoxyribose, are present at the carbon number 2 and the carbon number 3. In the sugar ribose, we have a hydroxyl group that is attached to the carbon number 2. This is the chemical structure of ribose. Now if you compare this structure with the structure of deoxyribose, the only major difference is at carbon number 2, that the carbon number 2 contains only a hydrogen atom and not the hydroxyl group. So the resultant sugar is called a deoxyribose. Here you can see the carbon number 2 in deoxyribose contains only a hydrogen atom, whereas in ribose the carbon number 2 contains a hydroxyl group. You can also tell this by the difference in their names, that the deoxyribose has a prefix called as deoxy, which means removal of oxygen. The resultant sugar, the deoxyribose, is more stable as compared to ribose because of one less functional group. The deoxyribose is present in DNA and ribose is present in RNA. You can also remember this by the starting of the DNA, which starts with D, so the deoxyribose, and the RNA, which starts with R, so the sugar is the ribose. The next important group to study in the structure of nucleotide is the phosphate group, which is this entity right here. The phosphate group consists of a phosphorus atom in the center to which 4 oxygen atoms are attached, which are negatively charged. The phosphate group is the same phosphate group which is present in the adenosine triphosphate, the energy carrying molecule of the body. However, in ATP we have 3 molecules of phosphate which are attached to one another by high energy phosphodiester bonds. The phosphate group is a polar molecule due to the presence of highly ionized oxygen atoms which impart negative charge to the phosphate group. Now the third important structure present in the structure of a nucleotide is the nitrogenous base. Nitrogenous bases are basically molecules that contain nitrogen in varying amounts and they act as a base. Now, most of you know that the human body contains organic chemicals which have carbon, hydrogen and oxygen in varying amounts. In nitrogenous bases, nitrogen combines with all of these atoms to form ring-like structures. These molecules are called bases since they can donate electrons to other molecules and form new molecules in this process. The nitrogen combines with all of these other atoms to form ringed structures. Now we can have two different types of ringed structures. One where there are single rings and second where there are double rings. The single rings are called pyrimidines and the double rings are called purines. We have three different types of pyrimidines: the thymine, cytosine and the uracil. And we have two different types of purines called adenine and guanine. In the center you can see the chemical structure of each of these bases. As you can see, the purines, which are double ring structures, adenine and guanine. Due to the presence of double rings, these purines are larger as compared to the pyrimidines. The thymine, cytosine and uracil all are single ringed structures and they are smaller as compared to the purines. The difference in the size of these bases is very important as it helps to pair these bases in the structure of DNA properly, which we'll study later. So now you know the individual structures which are present in a single nucleotide. Next we will look at how these individual structures bond to each other to form a single nucleotide, and then we will look at how many nucleotides bind to each other to form a chain of nucleotides, which is called a polynucleotide, which can be either DNA or the RNA. So if we take a sugar and name just the important carbons here, which will be the carbon number 1 and the carbon number 5, and of course you can see that at the carbon number 2 there is only a single hydrogen atom, so this sugar will be a deoxyribose. Now to pair this deoxyribose sugar with this pyrimidine ring, the bond formed will be between the N1 of the pyrimidine ring and the C1 of the sugar. If you want to go into the details about how these nitrogens are named, you can check the link in the description below for the IUPAC nomenclature of the aromatic compounds. Now this bond will be an example of a glycosidic bond, which is a covalent bond which links carbohydrates with other structures. Now if we have to pair a purine ring at a similar position, the bond formed will be between the N9 of the purine ring. Now next, let's attach the phosphate. The phosphate group attaches with the carbon number 5 of the sugar, and the bond formed is the example of ester bond, which is also a strong covalent bond. So now you know how the three structures in a single nucleotide bind to each other to form one nucleotide. Next, if we look at how many nucleotides bind to each other to form a chain of nucleotides called polynucleotides. So you can see basically what happens is that the phosphate group that attaches to the carbon number 5 of the sugar below also forms a bond with the carbon number 3 of the sugar above, and this pattern is repeated again and again. So you can see that the phosphate groups forms bonds both above and below the chain of these nucleotides. So as a result, a long chain of nucleotides is formed, which consists of a sugar phosphate backbone, and in the center we have all these nitrogenous bases which project from the sugar phosphate backbone. Now this can be an example of RNA if the sugar present in this example will be a ribose sugar. But if we take a similar strand of polynucleotide and run it in opposite direction, and this strand binds with the original strand through hydrogen bonds. Now here comes the role of complementary base pairing. Now what complementary base pairing says that only adenine forms a hydrogen bond with thymine and only guanine forms a hydrogen bond with cytosine. Now this is a universal rule that is applied in the structure of a DNA. So here you can see adenine forms a hydrogen bond with thymine, and similarly thymine forms hydrogen bond with only adenine. In similar case, guanine forms a hydrogen bond with cytosine and vice versa. Now this is the basic structure of DNA, and you can compare this with the step ladder structure of the DNA. You can see that the supports of the ladder in the red are basically representing the sugar phosphate backbone, and the rungs of the ladder represent the nitrogenous bases that are forming hydrogen bonds with each other. Now of course the original structure of DNA is that of a double helix, so you can compare this structure with a double helix where the helices of this structure represent the sugar phosphate backbone, and in the center you can see the red lines which represent the different nitrogenous bases that are forming bonds with each other.
So DNA is made up of 3 components. The first is a sugar known as deoxyribose. And it's called deoxyribose because there's a sugar ribose that has an oxygen right over here, but deoxyribose doesn't have that oxygen. So this molecule is deoxyribose, and the carbons in deoxyribose are labeled. This carbon is labeled 1 prime. Prime refers to that little apostrophe after the number. 2 prime, 3 prime, this carbon is 4 prime, and this carbon is 5 prime. And in case you're wondering why we need those primes, like why can't we just label the carbons carbon 1, 2, 3, 4, 5? So the answer to that question is that we're trying to differentiate between the carbons in this molecule. I will explain to you in a minute what this molecule is, but we're trying to differentiate between the carbons in this molecule and the carbons in the deoxyribose. So for some reason the carbons in this molecule took precedence, and the carbons there are labeled 1, 2, 3, 4, 5, etc. And so the carbons in deoxyribose are labeled 1 prime, 2 prime, 3 prime, etc. But anyway, that takes care of deoxyribose. And then the next molecule in DNA is a nitrogen base. And the nitrogen base you're looking at here is actually adenine. You may have heard of the molecule ATP, adenosine triphosphate, and that also has an adenine in it. But anyway, there are actually 4 different nitrogen bases that you can find in DNA. So I'm going to pause for a second from what we're looking at, and we're going to take a look at those 4 nitrogen bases. This one here is adenine. That's the base that we just saw a moment ago. And then right next to it, looking very similar, is another nitrogen base, guanine. And you can see that adenine and guanine are both double ring structures. On the left, you can see they have a ring with 6 sides to it, and then attached on the right, they have a ring with 5 sides to it. And well, these are all called nitrogen bases because they have a couple of nitrogens in them. And adenine and guanine are known as purines. So the double ring bases are known as purines, and I always have this hint to help me remember. So it's really an extrinsic hint because it has nothing to do with the material, but it always helped me. So when something is pure, it glows. So purines always glow. That was my hint, and then I would always remember that A stands for adenine and G stands for guanine. So if it helps you, you can use that. Then we have these other 2 bases. This one here is thymine. And then right next to it, we have something that also looks similar to it, cytosine. And you can see thymine and cytosine are single ring structures. They only have 1 ring with 6 sides, and they are known as pyrimidines. So again, the purines are adenine and guanine, and the pyrimidines are thymine and cytosine. And the purines and pyrimidines will always pair up with each other in this fashion. Adenine always pairs up with thymine, and guanine always pairs up with cytosine, unless of course there's a problem. And I want to just, let's just take a look at how these molecules pair up with each other. So let's look at this diagram. So the bonds that hold the nitrogen bases together are hydrogen bonds. So it's hydrogen bonding that puts them together. And let's just remind ourselves, hydrogen bonding takes place in molecules that have a hydrogen attached to one of 3 very electronegative atoms, fluorine or oxygen or nitrogen. Let me remind you, electronegative means that they like to hog electrons, they pull electrons towards themselves. And what's going to happen in molecules like this is that since fluorine or oxygen or nitrogen hog electrons, they are going to get a slightly, or maybe more than slightly, negative charge, which leaves the hydrogens kind of bereft of electron density, and gives them a positive charge. And then the molecules will orient themselves in a way where the positive and negative sides are attracted and attached to each other. So let's actually take a look at what I just explained in the molecules. So let's look at thymine and adenine. So we have this oxygen over here, which is going to be somewhat negative because it's pulling electrons away from that carbon and in this double bond. And then these hydrogens are going to be somewhat positive because the nitrogen near them is pulling electrons away. And so they form this hydrogen bond right over here. Then we have another hydrogen bond between this positive hydrogen. Remember, it's positive because the nitrogen near it is very electronegative and hogs all the electrons. And then we have this negative nitrogen because it hogs electrons from the carbons around it. So between thymine and adenine, we're going to have 2 hydrogen bonds. But if you look at cytosine and guanine, there are actually 3 hydrogen bonds between them. So we can say that cytosine and guanine are attached to each other a little bit more strongly than thymine and adenine.
In this video, we will study about the structure and function of the DNA. DNA stands for deoxyribonucleic acid, and DNA is a molecule which is located inside the nucleus of the cell, and its main job is to store and code the genetic information of the body. Now, for most of the life cycle of the cell, the DNA is located inside the nucleus of the cell like a coiled noodle. But during the time when the cell replicates, the DNA is arranged in the form of structures which are known as chromosomes. And these chromosomes help to keep the DNA stable during the time of the cell replication. Now, most of you are familiar with the double helix model of the DNA. The double helix model demonstrates that DNA is basically a polymer. Now, to understand the structure of DNA in a better way, you have to understand the definition of a polymer. So the polymer is basically a molecule which is consisting of many repeating units which are known as monomers. If we take the example of a simple polymer called as starch, in this case the repeated monomeric units are known as glucose. These glucose molecules are joined together by glycosidic bonds and they form the polymer which is known as starch. In a similar way, DNA is also a polymer, and in this case the monomeric units which form the polymer DNA are known as nucleotides. Now, this is the basic structure of a nucleotide. You can see that it consists of 3 important groups: the phosphate, the sugar, and the nitrogenous base. Now, if you want to study the details of the structure of nucleotides, you can refer to the video about the nucleic acids. The link to that video will be in the description below and also on the top of the video. But in a nutshell, you can conclude that the sugar present inside the nucleotide is called the deoxyribose, which is a 5-carbon sugar which belongs to the pentoses. The phosphate group consists of a phosphorus ion to which 4 oxygen atoms are attached. And then we have the important nitrogenous bases, which are organic molecules which contain nitrogen in the form of ringed structures. We have 4 different types of bases that can be present inside the molecule DNA. These are the adenine, guanine, cytosine, and thymine. Now, since we can have 4 different types of nitrogenous bases, we can have 4 different types of nucleotides that are present inside the structure of a DNA. Now, as you can see, these 4 nucleotides are arranged in 2 groups. The first group is known as pyrimidines, and these contain the nitrogenous base thymine and cytosine, which are both single ringed bases. And the second group contains the nucleotides, which are known as purines, which have adenine and guanine as the nitrogenous base, which are double ringed structures. Now, in case of starch, the structure is pretty simple, which only consists of long linear chains of glucose or either branched chains which are joined together by glycosidic bonds. This is the chemical structure of the starch, and you can see the different types of glucose chains. But in case of DNA, the 4 types of nucleotides join together in a very complex way to form the double helix structure. Now, if you zoom into the structure of this double helix model of the DNA, you can clearly see that it basically consists of these 2 blue lines which are twisted around each other, and it also consists of these orange lines in the center which connects these 2 blue lines together. Now, to understand its structure in a better way, if we just untwist the double helical model, we will get something which is known as the step ladder model of the DNA. It is also has a similar structure, and it consists of these blue vertical lines and these orange horizontal lines. Now, these blue vertical lines basically represent the sugar phosphate backbone of the DNA. And the central lines, which are orange in color, represent the base pairs of the DNA. We will get to these terms in just a bit. Now, the carbons present inside the deoxyribose sugar of the DNA are numbered from 1 to 5 in the clockwise direction. And you should remember this numbering of the carbon atoms because it will help you to understand the concept of directionality of DNA. Now, what happens is that the third carbon in every deoxyribose sugar forms a bond with the phosphate group of another nucleotide that is present below it, and this bonding extends in both directions. Now, if you zoom into this structure, you can basically see that every phosphate is connected to two sugars, one bond with the third carbon of the sugar above and one bond with the fifth carbon of the sugar below. And now again, if we zoom out, you can clearly see that due to the bond formation between the sugar and phosphates, we create this sort of sugar phosphate backbone on one side, and we have all the nitrogenous bases which project out of this sugar phosphate backbone on one side. So what this basically represents is essentially a single strand of DNA. Now, if you go back to the step ladder model of the DNA, you can clearly understand how the different nucleotides in a single strand of DNA are joined together to form the sugar phosphate backbone, which was represented by these blue vertical lines. And in the center, we had these orange bars which connected these two lines, which are essentially the base pairs from one side. Now, in the opposite side, we have the other strand of the DNA, which is quite similar to this original strand, but has some key important differences for you to understand. And to understand these key differences, you have to understand the concept of directionality and complementary base pairing. Now, what directionality essentially means is that it is the end-to-end chemical orientation of a single strand of nucleic acid. In this example, you are saying the single strand of a nucleic acid. Now, the direction of the DNA is expressed in two ways, 5' end to 3' end or 3' end to 5' end. Now, if you look closely at this structure, you can basically see that it has a 5' end to which a phosphate group is attached, and it also has a 3' end below to which a hydroxyl group is attached. So if we come back to our DNA ladder model, we can see that the strand we made before runs in 5' to 3' direction. Now, in a similar way, if you look at the opposite strand, you can basically note that there is one important key difference, that it runs in completely opposite direction. It has its 3' end in this direction and its 5' end in this direction. So it is said to be running from 3' to 5' direction, which makes the two strands of the DNA anti-parallel. Now, the next important thing for you to understand is the complementary base pairing, which describes the manner in which the different nitrogenous bases of DNA strands align with each other. So it basically means that the base adenine pairs always with the base thymine, and the base guanine always forms hydrogen bonds with the base cytosine. You can see this rule in action in the DNA stepladder model, where you can see that the base A always pairs with the base T and the base C always pairs with the base G. These bases are essentially forming the hydrogen bonds in the center, which holds these two strands of the DNA together. Now, the complementary base pairing is very important in one of the most important aspects, that it maintains a proper distance between the two strands of the DNA, which is very important for the stability of the two strands of the DNA. So now if you look at the double helical model of the DNA, you can understand what these two helical lines mean and what these central spokes which are connecting these two lines are essentially made up of. So this was all about the basic structure and function of our DNA.
As long as human beings have been around, I could imagine that they've noticed that offspring tend to have traits in common with the parent. For example, someone might have told you, hey, you walk kind of like your dad, or your smile is kind of like your mom, or your eyes are like one of your uncles or your grandparents. And so there's always been this notion of inherited traits. But it wasn't until the 1800s that started to be studied in a more scientific way with Gregor Mendel, the father of genetics. But even then, even Mendel, who was starting to understand the mechanisms of, or he was trying to understand how inheritance happens, and he even could start to breed certain types of things, even he didn't know exactly what was the molecular basis for inheritance. And the answer to that question wasn't figured out until fairly recent times, until the mid-20th century, not until the structure of DNA was established by Watson and Crick. And their work was based on the work of many others, especially folks like Rosalind Franklin, who essentially provided the bulk of the data for Watson and Crick's work, Maurice Wilkins, and many other folks. But it was really the structure of DNA that made people say, hey, that looks like the molecule that's storing the information. Just to be clear, DNA wasn't discovered in 1953. DNA was discovered in the mid-1800s. It was this kind of this molecule that was inside of nuclei of cells. And for some time, people said, oh, maybe this could be a molecular basis of inheritance. You know, you can imagine what you would need to be a molecular basis of inheritance. It would have to be a molecule or a series of molecules that could contain information, that could be replicated, that could be expressed in some way. But it wasn't until 1953 when this double helix structure of DNA was established that people said, hey, this looks like our molecule. So first, let's just talk about the structure here, and then actually we'll talk about where this name DNA, deoxyribonucleic acid, comes from. And then we'll talk a little bit about why the structure lends itself well to something that stores information, that can replicate its information, and that could express its information. We might go in depth on the expression of information in future videos. So this structure right over here, and this is a visual depiction of a DNA molecule, you can view this as kind of a twisted ladder. It has these two, I guess you could say, sides of the ladder that are twisted. That is one side right over there, and then it is another side. There's another side right over here. And in between those two sides, or connecting those two sides of that twisted ladder, you have these rungs. And these rungs are actually where the information, the genetic information, is, I guess you could say, stored in some way. Because these rungs, it's a sequence of different bases. And when I say bases, that you say, wait, this says acid. Why are you saying bases right over here? Well, the word deoxyribonucleic acid comes from the fact that this backbone is made up of a combination of sugar and phosphate. And the sugar that makes up the backbone is deoxyribose. So that's essentially the D in DNA. And then the phosphate group is acidic, and that's where you get the acid part of it. And nucleic is, hey, this was found in nuclei of cells. It is nucleic acid, deoxyribonucleic acid. But it's not, it is actually mildly acidic all in total, but for every acid, it actually also has a base. And that base, those bases form the rung of the ladders. And actually, each rung is a pair of bases. And as I said, that's where the information is actually stored. Well, what am I talking about? Well, let me talk about the 4 different bases that make up the rungs of a DNA molecule. So you have adenine. Adenine. And so, for example, this part right over here, this section of that rung might be adenine. Maybe this right over here is adenine. This right over here. Remember, each of these rungs are made up by, it's a pair of bases. And that might be adenine. Maybe this is adenine. And I could stop there. Maybe I'll do a little more adenine. Maybe that's adenine right over there. And adenine always pairs with the base thymine. So let me write that down. So adenine pairs with thymine. Thymine. So if that's an adenine there, then this is going to be a thymine. If this is an adenine, then this is going to be a thymine. Or if I drew the thymine first, well, I'll say, okay, it's going to pair with the adenine. So this is going to be a thymine right over here. This is going to be a thymine. If I were to draw this, this would be a thymine right over here. Now, the other 2 bases, you have cytosine, which pairs with guanine, or guanine that pairs with cytosine. So guanine, and we're not going to go into the molecular structure of these bases just yet, although these are good names to know because they show up a lot and they really form kind of the code, your genetic code. Guanine pairs with cytosine. Guanine and cytosine. So actually, if this is, let's say there's some cytosine there, let's say cytosine right over here, maybe this is cytosine, maybe this is cytosine, maybe this is cytosine, this is cytosine, hey, maybe this is cytosine, then it always pairs with the guanine. If we're talking about, so let's see, this is guanine then this will be guanine, this is guanine, this is guanine. I actually didn't draw stuff here, but this is guanine. I didn't say what these could be, but these would be made of pairs of, they could be adenine-thymine pairs, and it could be adenine on either side or the thymine on either side, and they could be made of guanine-cytosine pairs, where the guanine or the cytosine is on either side. Actually, just to make it a little bit more complete, let me just color in the rungs here as best as I can. So those are guanine, so they're going to pair with cytosine, pair with cytosine, pair with cytosine. And when it's drawn this way, you might start to see how this is essentially a code. The order of which the bases I guess the order in which we have the sequence of these bases essentially encode the information that make you you. And you could debate, well, how much of it is nature versus nurture? And when people say nature, it's literally genetic, and that's an ongoing debate. But it does code for things like your hair color. When you see that your smile is similar to your parents, it is because that information to a large degree is encoded genetically. It affects a lot of what makes you you, and actually not even just within a species, but also across species. Humans have more genetic material in common with other humans than they do with, say, a plant. But all living creatures as we know them have genetic information. This is the basis by which they are passing down their actual traits. Now, you might be saying, well, how much genetic information does a human being have? And the number will either disappoint you or you might find it mind-boggling. The human genome, and every species has a different number of base pairs, to a large degree correlated with how complex they are, although not always. But the human genome has 6 million. Sorry, not 6 million, 6 billion. 6 million would be disappointing. Even billion might be disappointing. 6 billion base pairs. 6 billion base pairs. And when you have your full complement of chromosomes, and this is in most of the cells in your body, and outside of your sex cells, the sperm or the egg cells, this is going to be spread over 46 chromosomes. 46 chromosomes, or I guess you could say 23 pair of chromosomes. So if you divide 6 billion by 46, you get a little over, on average, 100 million, I think it's 100 and something million base pairs per chromosome. And some chromosomes are longer, actually some of the longest are over 200 million, and some might be shorter. That's just on average. Now this number might, to some of you, might be exciting. You're like, oh, I thought I was a simple creature. I didn't know I was this complex. 6 billion, that's a lot of base pairs. That feels like a lot of information. For others of you, it might not feel so great. You might say, hey, wait, I could store this much information on a modern thumb drive or on a hard disk. I thought I was more unique than that. And of course we all are special and unique, but you might say, oh, 6 billion base pairs, I thought I was infinitely complex and whatever else. And there's some arguments for that in along some other directions. But this is the approximate length, I guess you could say, the approximate size of the actual human genome. And when we talk about chromosomes, and we'll talk about chromosomes in much more depth, imagine taking this zoomed in thing that you have right over here. And over here, I don't know how many we have, like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. We have about 20 base pairs depicted here. Imagine if you had about 200 million of these base pairs, and then you were to take this thing and you were to kind of coil it up into, that thing is a chromosome. Is a chromosome. And you're saying, wait, I have that much information in most of the cells of my body? This thing must be incredibly compact. And if you said that, I would say, yes, you are correct. The radius of the DNA molecule is on the order of 1 nanometer. 1 nanometer, which is a billionth of a meter. So you can start to assess kind of the scale of this thing. This is a very dense way to actually store information. But just to have an appreciation of, and you might have seen it when I was coloring in, on why the structure lends itself to being able to replicate the information or even to be able to translate or express the information, let's think about if you were to take this ladder and you were to just kind of split all the base pairs. So you just have 1/2 of them. So you essentially have half of the ladder. And so if you only have half of the ladder, you're able to construct the other half of the ladder. Let's take an example. Let's say, and I'll just use the first letter to abbreviate for each of these bases. So let's say you have some, so let's say this is this is the sugar phosphate backbone right over here. So this could be one of the sides. And let's say there's some adenine. Actually, let me do them in the right color. So you got some adenine, maybe some adenine right over here. Maybe there's an adenine there. Maybe you have some thymine, maybe thymine right over here. Then you have some guanine, and then let's say you have some cytosine and you have some cytosine. So if with just half of this ladder, I guess you could say, you're able to construct the other half. And that's actually how DNA replicates. This ladder splits, and then each of those two halves of that ladder are able to construct versions of the other half, or versions of the other half are able to be constructed on top of that half. So how does that happen? Well, it's based on how these bases pair. Adenine always pairs with thymine if we're talking about DNA. So if you have an A there, you're going to have a T on this end, T on this end. T's right all over here, T right over there. If you have a T on that end, you're going to have an A right over there, A. If you have a G, a guanine on this side, you're going to have a cytosine on the other side. Cytosine. And if you have a cytosine, you're going to have a guanine on the other side. And so hopefully that gives you an appreciation of how DNA can replicate itself. And as we'll see also, how this information can be translated to other forms of either related molecules, but eventually to proteins. And just to kind of round out this video, to get a real visual sense of what the DNA molecule looks like, or I guess a different visual depiction from this, I found this animated GIF that, you know, if you haven't fully digested what a double helix looks like, this is it. And you see here, you see your sugar phosphate bases here. You see kind of the sugars and phosphate, the sugars and the phosphates alternating along this backbone. And then the rungs of the ladder are these base pairs. So this is one of the bases. That's its corresponding, I guess you could say, partner. And you could see that along all the way up and down this molecule. Very exciting.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.